Well, hello and welcome to Leading Minds, expert voices from the College of Medicine, Nursing and Health Sciences at NUI Galway. My name is Jonathan McCrea and I'm delighted to be your host for this series. The aim of the college is to improve healthcare locally, nationally and internationally. And in this episode, we're going to meet Professor Garry Duffy, who's a professor of anatomy and regenerative medicine and vice dean for research and graduate studies here at the College of Medicine at NUI Galway. Garry, you're very welcome to the podcast. Thanks for joining us. Perhaps you might start off by telling us what your area of research and interest is at the college.
Thanks, Jonathan. Yeah, our main area of interest at the moment is around the foreign body response to medical implants, and medical implants that go into the body for different reasons. And what the body does when it goes in at the first time is it actually sees it as foreign and tries to responds to it, and then tries to change the dynamic of the interface between the device and the body. So a good example of this is that when you put in a device like an implant, say it's a silicone-based implant, the body will actually respond and wall it off in reality in trying to keep it separate from the tissue. And it does this because it sees the material properties or the stiffness or the compliance of the material as foreign, and then it doesn't want to integrate it into the body itself.
How does it know that this thing isn't part of the body?
So it's like a normal response to any kind of foreign element that goes into the body. If you kind of think of bacteria or viruses, the immune system generally knows, okay, that's not correct. That's not the same as what the surrounding tissue is. And there's certain cell types that come in first to check it out. So you get these neutrophils first. They call on the macrophages, which are the engulfers. They kind of take things and eat it up and get rid of it. So those macrophages come in, and the first thing they try to do is actually eat the device in reality, but it's too big. They coalesce with their other macrophages and they form these giant cells. These giant cells try and engulf. And then if that doesn't really work, they start to think, okay, we have to call in other cells to wall this off. So these other cells come in called fibroblasts, and they start to lay down a matrix around the device to wall it off completely. And we're really interested in understanding that dynamic, understanding why it happens, how it happens. And we're really interested in to try and figure out, can we reduce it? Because then you may have longer lasting implants in the body, or implants that don't cause as many issues when delivered.
It's really interesting to hear the various stages of defense that our body has against something that shouldn't be there. What are your approaches to trying to make the body accept these implants, which are often crucial for the survival or the well-being of the patient?
Yeah, it's exactly that. And most implants have to have a function, right? And form normally follows function. But the big thing for us is to try and make things more compliant. So a lot of previous implants would be very rigid. They would be made of certain plastics. They would have a defined form. They're delivered in a certain way. But now we need more complex implants because they need to interact with the body, the biological system. They're not just there like a knee replacement to replace knee function. These are integrating with the body in terms of providing a particular function. So now we have to try and think about making them either more compliant, choosing materials that don't elicit this foreign body response. And a lot of what we do is actually try to understand the foreign body response first and then come around. And a big area of interest for us right now is the soft robotics idea of using compliant materials, or materials that have similar properties to body's tissue, particularly soft tissues like muscle, and connective tissue around muscle.
I find that really interesting because I would have thought that the way the immune system identifies foreign bodies would have been solely around the molecules and chemicals and materials on the surface of the object. But you're saying the actual rigidity and shape of the object can also trigger an immune response. How do you go about investigating these things? Because you can't just put random things into people and see what happens. How do you investigate that?
Yeah, so exactly the mechanical properties, that stiffness, roughness is another example. It's not generally a material property, but it's it's something that's more characteristic materials. They can elicit the response. The good example I use of this is that how does a cell know that something is stiff? It basically attaches to it and then tries to contract. And if it can't contract, it can't pull on its ends, then the material is too stiff. So we look at a lot of in vitro models or on the bench models with different cell types, where we look at that response. So we look at how does the cell integrate? Does it attach? If it attaches, does it try and shorten? If it can't shorten, does it start to release factors that tell other cells to do things? And then we have other assays that are looking at, deposition of material. So like the cell will respond by producing biological material, particularly collagens. And those collagens, then we know, okay, if there's an increase in collagen, the cell doesn't like this particular material. It kind of sees it as foreign. So there's a whole area of not just the chemical structure, the material property, that mechanical property actually seems to play a lot more. So that kind of compliant materials is where it's becoming more exciting, because you can actually change a material and it can change its stiffness by actuating it. And I just use the example of inflating and deflating it as a good example. I've changed the stiffness property of the material. It's the same material, but it's got a different mechanical property. And then the body might see that as different. That's the general reality.
So you're putting on different disguises, I suppose, onto each of these devices to see which one gets past the body guards. Can you tell me of the different approaches there? Because obviously one way of doing it might be to dampen down the distress calls or the factors asking for recruiting other types of cells. Another way might be to make something that is covered in human cells to start with. What are the various approaches to overcoming this foreign body problem that you're looking at?
Yeah, so in our own lab, we've kind of broken it down into this idea of, bioinspiration of, like, if you kind of think of a defense mechanism and then a prey, and we kind of think the immune system is the prey, and then what defense mechanisms can we take from, biologically inspired. So there's a number, like camouflage, as you mentioned, is one that's used a lot in the animal kingdom. Changing shape, we use the example of the puffer fish a lot, like it basically inflates itself. A similar scenario we're kind of thinking of from these actuating devices. Or exactly as you described, they can release chemicals. There's often animals in the animal kingdom that release chemicals to ward off any type of prey. So we can kind of think of that exactly as you described. The one that we're really interested in right now is not to add something else in, because the material is already foreign. We want to actually tune the material properties over time, because that for us is quite simple. And so we kind of take that idea of movement being something of interest, fluid flow across the device being very interesting. And that kind of comes back to that idea of the puffer fish again. And then the other thing we think about a bit is that the other defense mechanisms are physical. So when there's this capsule surrounding a foreign body, you could actually pierce it, right? You could pierce it and allow things to leave it then. So again, coming back to that kind of idea of, like, a puffer fish has got spikes on the outside and they're evident when the prey tries to eat it. Exactly that idea. We leave a device, static for a period of time, and then we basically spring out some needles on the surface that would then kind of poke holes in the capsule. So that's the general principle. So and it's kind of interesting to work on, or kind of fun to work on, because you get to kind of think of those that, movement can be simple, as in, inflate, deflate, but it can be twisting, it can be bending. There's all of these other elements that you can bring in to the design process.
That's what you mean by actuate.
Actuate. Yeah. And why we use that word actuate and instead of inflate and deflate is that actuation could be, pneumatically air driven. It could be liquid driven. It could be, an energy response of the material. So like if you put an electric current to it, it changes its shape. So the overall word is actuation, but it can be driven by different mechanisms and control systems.
So are there certain parts of the body which are much more prone or much more likely to reject foreign bodies than others? Because we have an approach for some implants that work quite well, right? And they stay there for long periods of time. People get plates in their knees or whatever, and some people get pacemakers and so on. Why do those things work, and why are we having problems with other types of things?
Yeah, it's definitely the blood contact piece that becomes important, right? So, the immune system is generally in the blood, if you kind of think about it, and the lymphatic system. So anything that interacts with that kind of liquid interface piece in the body, that's going to be somewhere you may see. Like pacemakers are a good example of that. We do have good longevity, but even in the first week of implanting, the best pacemaker that's out there, the signal will reduce from that pacemaker into the tissue, the electrical signal across. So we do have the foreign body response to that, but there is ways that, you put more energy through that then to generate the functional response you have. So that's how we're responding to the foreign body. It's not that the pacemaker doesn't have it. It's just that, you can turn up that signal a little bit on the pacemaker to kind of make it work a little bit better. A big area is like deep brain stimulation. I would see that as a major advent in the Parkinson's arena. That definitely shows a lot of the foreign body response, particularly in the nervous system. Right. And then anything that's going in around muscles or any kind of soft tissue, again, it's generally, they don't integrate with the muscle. The muscle wants to still have its activity, so they're walled off separately to it, if that makes sense.
There's also different types of functionality of these sort of implants that you're looking at, right? Because delivering electrical pulses is one thing, protection or strengthening of bone joints is another. But when it comes to drug delivery, that's a really exciting area that you're looking at. Maybe you might tell me a little bit about that and a little bit about how these devices of the future need to be more interactive.
Yeah, so like I give a good example of, a patient who has type 1 diabetes, or a person living with type 1 diabetes, they deliver insulin themselves, and there's different ways to deliver insulin. One form of that is a cannula, and it's connected to a pump. And a cannula is just a very, very small tube that goes subcutaneously under your skin. But you have to change that regularly because of the foreign body response or kinking. There's other problems that can happen to it, but foreign body response is one of them. Because you need that tube to be open for the insulin to pass through it to get into the body for it to work. So we're really interested in that because, at the moment you change them, between 3 and every 5 days. So if you kind of think of that for a person living with type 1 diabetes, that's a lot of device changes over time. And then you have to have a particular site that you're going to put that in your body, and it's, the abdomen or the back of the arm, or so we're really interested in understanding, okay, what is the foreign body response to that cannula, first of all, and then can we use our soft robotics approaches to reduce it, for instance, or overcome it? And that's one of the other things that we've tried to open our mind to, is that the foreign body response we can see as negative, but we can also see it as an opportunity for device integration into the body if we get it right. So in that particular drug delivery space, we need the drug to get out. So we do a lot of work in that right now, where we're trying to figure out, okay, what's the failure? How is this failing? Why is it failing? And then how can we design devices to overcome that level of failure? And we basically want a device to be implanted for 30 days. So instead of changing it every 3 days, you're changing it 12 times a year. The other version of that is that we can actually implant it. So instead of it being a cannula that's just going into your skin or subcutaneously, we're thinking of, okay, one other advent of this is an implant that lasts for a year, that you need implanted for a year, and then you don't have to have any changes over that year. But there's a bit of a step change in the kind of clinical reality of going from something that's put in through your skin to something that's implanted. That's not a simple move. But again, if we generate the device well enough, then we can kind of think of, okay, we can do that. We can actually put it in. And then like a lot of our work then is around how do you get those devices into the body in a minimally invasive way, something that could be done as a quick procedure in a healthcare office kind of thing, a GP's office as an example, or not that it requires surgery for implant.
Yeah, it'd be great if we had just sort of flaps we could open and take things out. So the implantation of these devices, obviously that's an end goal. You said a year. Would it not be possible to create something that would last 15 or 20 years and deliver drugs? Or is that just, is it a very difficult thing to do, to have enough of the drug you need in there, or for it to be guaranteed to last that long? What's the problem there?
Yeah, the guarantee to last that long is probably the bit, because the device integrating with the body might work, but for you to be able to fill it with drug, you have to have a port. So there are issues then with port failure, and like we know a lot of this from patients who are having chemotherapy, and they have a central line put in, and then that central line is used for the chemotherapy delivery. There are failure modes around that port, that port sitting outside the body. So it's that refill piece for us that we don't want to try and do something that's 5 and 10 years. We'd love to get there, but we need to prove it well in a year fashion first, get it down to a minimally invasive delivery, make sure it works correctly, there's no failures at the port, and then it could be successful. And if we get it right, I don't think there's any reason it can't be longer. But for us, a good period of time right now is 12, because we can test that as well in the models that we have.
What do you make these soft robots from? And are there benefits to them being soft as opposed to rigid, apart from the ability to fool that foreign body response?
Yeah, so as part of our type 1 research work, we did a lot of work with people living with type 1 diabetes. We did what we call patient panels. We did them in Ireland, in Germany, and in Spain. And what we got feedback on is that people don't want to feel an implant in their body. The rigidity of an implant in their body And we didn't actually, that was a surprising feedback to us in general. So that's one thing that it gives. It gives you a compliant material that's not felt as rigid from outside the body. So then that's good. It can integrate and quality of life is improved. And then the other thing is that it moves in a similar way. Then you don't kind of have this, your body is soft and then your device is rigid. And over time, there's friction and issues with it. They'll actually integrate better with each other when they're soft. So it's kind of that long-term, implant function is what we're really trying to get at.
And so silicone obviously is
silicone. Yeah, polyurethane. We do a lot of it, like polyurethane. There's particular type of material processes that we have to get right. So a big area we like to work on is with polyurethane, and we use a method called thermoforming, so we can form over different materials. So we need the polyurethane to be thermoplastic to respond to temperature and to allow us to stretch it. And that then allows us to create devices of complex geometries, that it's not just a simple tube, it's something that's wider on one end and skiddier on one end. And then silicone, we've done a lot of work with Ona Carroll's group in UCD on additive manufacturing approaches to silicone devices. So again, it's a great material. There's lots of evidence about it being quite useful for implants in the body. I know there's also negative aspects when we talk about breast implants as one example. There's been some issues associated with some rare cancers and some of the material choices there. But again, it's something that we're really looking at to try and understand. They'd be our top choices right now. And then are there other materials? And then what we coat those with is probably another thing that can be a little bit new and novel.
Where do stem cells and cellular matrices fit into all of this? Like, could you, for example, create a stream of stem cells by using one of these soft devices to help with certain conditions, or could you use stem cells to coat these objects to make them more friendly?
Yeah, so there's kind of two areas to that we're interested in. Is that the foreign body response in general? There's MSCs or mesenchymal stromal cells have been shown to reduce the immune effect to treatments in general. So there's a lot of work going on in co-delivery of MSCs at the time of implant to reduce the overall foreign body response. For us, we're really interested, and I come back to the type 1 example, is that we're interested in insulin delivery because that's the current treatment. But we believe in the future that stem cells and stem cells that can release insulin will actually be the future treatment for people with type 1. So we're very interested in how do we get them into the body and how do we keep them okay in the body for long periods of time. So it comes back to that device again. We're going to put them inside a capsule or a bag is the best example. And that bag has to stay in the body for a period of time. So again, we're looking at the foreign body response to that. And what we'd like then is that has a long-term implant. It is a long-term implant. You could refill it with more stem cells if the stem cells get exhausted, but the device stays there for a period of time.
Nice.
So what we've been thinking about is how do we make that long-term, and we get back to that actuation piece. So before we fill it with stem cells, we actuate it and try and reduce the foreign body response and then fill it with stem cells. So it's kind of that idea of how do you make the implant the best implant? Yeah.
This sounds like a stupid question, I'm sure, to you, but do we have lots of space in our body that we can stick things in? I mean, when I think of my body, I like to think of it as like something very compact, of course. But I do like to think that there aren't that many holes lying around. Could we just stuff stuff in between organs and it would be fine?
Yeah, so as an anatomist as well, we kind of look at this whole idea of what are the potential spaces in your body. I completely agree. The abdomen is a great spot that's compliant, right? So it's an example of you can eat a lot of food one day and not eat a lot of food the other day, and your abdomen doesn't really change shape. So the idea that there is space available and your abdominal contents will give up a little bit of space for devices. But that's also a kind of a surgical risk because you're putting it into an environment that could also cause things like peritonitis. So where the actual abdomen responds not only with foreign body response, but the whole thing gets inflamed. So we're really interested in what we call the anterior abdominal wall, the front of your abdomen. There's layers of muscles there, and there's generally 6 layers across your abdomen. You generally know about the six-pack muscles, the rectus abdominis. We know about those quite well. Yeah. But between those, there are potential spaces, and they're not spaces now, but we can make them into spaces. So we also look at that quite closely in our group and with our collaborators on how can we create a potential space between two layers. So what we do is we inflate a balloon and we push the two layers open. That's how we create the space. And then what can we put in there? So we don't want the device to be huge. You don't want to be like putting a Coke bottle between those two layers. So we're going to try and pick a layer that can accommodate a certain volume in a very, like a flat fish kind of point of view is the way I describe it. You put it in between the two layers. And then that compliant material piece is really important because your abdomen works for your posture, turning, sitting, your crunches, all of those. So whatever we put in there needs to fold and bend and twist with that same area. So we've done a lot of work on identifying spaces in the anterior abdominal wall and how do we deliver into them. So the delivery tools that we put in. And that's kind of an exciting area where we're now looking at the patient population there. So we've linked in with experts in the University of Michigan, where they've given us access to their what they call their analytical morphomics database. So really, what does that mean? Morphomics is just like what is the shape and space of things, and then analytical means we're not just looking at one, we're looking at a lot. So we were able to get access to about 600 CT scans of abdomens of people who would have type 1 diabetes, and to look at these spaces and to map them out. And we use very defined tools to do that, a little bit of machine learning to understand, could we use some bony landmarks to tell us what size of a device would fit into a person. And the idea there is that someone would go to their GP, the GP would measure from the bottom of their ribs to their pubic bone, and they would measure out to the top of their hip bones, the iliac crest, and then that would help them define what device size would fit into their abdominal wall. So yeah, and that's been a really
fitted for a tailor or something.
Yeah, it is a little bit because it has to be bespoke. And the good example that we kind of give is there's also your right and left side of your abdomen are often different. If you're right-handed, it's bigger on the right-hand side than the left-hand side. The spaces now, not, you wouldn't notice this in your own abdominal wall, but when you look at the more detailed anatomy, the spaces are different. So we also wanted to pick a site that if we're putting a device in, we have the option to put a second device in if someone grows or if the first one doesn't work out as well. So why we picked the abdominal wall is if you kind of think of it, it's got a line down the middle, and then you've got spaces on either side of that line, the linea alba. So we're kind of looking at that space a little bit more. And like we found that there are spaces that can hold enough cells to reverse type 1 diabetes in general, or to reduce the need for amounts of insulin. The patient may not become insulin independent completely, but they may reduce their dose of insulin over time, or their frequency, or how much time they have to check their blood glucose, etc. So we're kind of excited about that, and we need to expand it, beyond the 600 scans that we've looked at and gone through. But in those scans, we went right from young patients, 8 years old, all the way up to people that are 65. Because again, the idea of this space is the same and everyone is incorrect. We have to design correctly for the spaces that are the patients.
I suppose the breadth of experience and the different colleges in NUI Galway, presumably that's a huge benefit because of the expertise that lies in engineering, in stem cells, in foreign body response. I presume the sort of work you're doing stretches across a whole lot of different disciplines.
Yeah, and I'm probably speaking as if we do a lot of this work on our own. That's not the case at all, and I think that's exactly the advantage of the College of Medicine and Nursing and Health Sciences, as you described, gives us access to those clinical experts. So Derek O'Keefe and Tomas Griffin and Aoife Lowry from the surgical point of view has given us great advice on how you'd approach this from an interventional point of view and a patient point of view. Emer Dolan in engineering, an expert design engineer, has helped us figure out, okay, how can we make a device that fits in here? And she's leading her whole program on islet transplant now. Barry McDermott did all the machine learning for us. He basically loves taking a complex problem and understanding it at that detail. And then, Scott Robinson, who was a research fellow in a lab, he was a surgical trainee, has now gone back to Michigan to finish his surgical training, and now in Florida. So it's also not only a Galway scenario, it's a bit of a link. And then it's probably important on the soft robotics. We link really closely with Ellen Roach. She's a professor at MIT. She's also, Irish and a Galway graduate as well. And that's been really good for us because we've had staff exchange. So, they run some of our studies, we analyze their technologies. And then from the anatomy point of view, we have the advantage of, looking at human anatomy But also then anatomy generally comes with microscopy, right? That ability to image. So we've been lucky that the team here and our head of department, Peter Dockery, he's been able to help us, answer some of these what's going on at the interface. And a good example or a story I tell people is that, when Scott was here with his surgical background, he didn't like to like cut sections all day long and like sit there and look at, hundreds of slides. So he developed a method to actually take the whole sample, stain the whole sample and image the whole sample using CT and MRI. Because again, we get the same information. So now we've developed a new method that, isn't as labor intensive as well. So there's that kind of input knowledge that has to happen. And then obviously on the immunology side, I wouldn't say I'm an expert in immunology. So we definitely have to link now that we know that we can modify the foreign body response. We know it works. We've shown it multiple times. We still don't know how it's working. That's the bit we're at. So I'd say for the next 3 to 5 years, the group is going to get into that space a little bit more and link with colleagues across Ireland and NUIG on that, because it's not straightforward. Like I said 2 cells earlier, and it's probably 30 cells that are involved in that initial response.
I suppose it's great to know that over the past 2 years we've grown a whole country of immunologist experts. So quite cool to draw from. Really brilliant speaking with you. Such exciting work and really innovative as well. I can't wait to see where some of these ideas take us in 10 or 15 years. Professor Garry Duffy from the College of Medicine, Nursing and Health Sciences at NUI Galway. Thanks as always.
Thanks, Jonathan.
Well, thanks for joining us. I hope you enjoyed this episode. You'll find lots more in the podcast feed wherever you found this podcast. I'll see you next time.